Without further ado, Marcel, thank you for joining us, and we look forward very much to hear through your eyes what's the business case for comprehensive genomic profiling in your lab. Thank you.
Thanks very much for the very kind introduction. I keep on going, and I start from here. So as first indication, or as first feeling to our clinical setup, we are a very stereotype sized department of pathology, mainly focusing on FFP origin material to be analyzed by standard traditional technologies, being immunohistochemistry-ish or genomic profiling based mainly on DNA or RNA-seq technology. So we have focusing on NGS, our NGS core genomic platform. We have an Illumina-driven setup, which is comprised by semi-automated DNA, RNA isolation platforms and robotic system, which we in the meantime implemented, and multiple Illumina sequencing platforms from a smaller sized benchtop mini-seq to a now just ordered NovaSeq X scale. We started in 2019 with a TrueSight 500 CGP option being available on the market in connection to the PRNDX clinical reporting option to choose from. For multiple reasons, which we are going to discuss, we shifted based on our experience and the offerings OncoDNA is providing with the OncoDEEP Kit in the late of 2022 into the validation and evaluation of the CGP option OncoDNA is offering. And we implemented the automation of the whole library prep in 2023, and now are running it for almost more than 2 years in our routine clinical diagnostic setup. When we have a look at the dynamics concerning the DNA-based NGS panels, of course, we are all familiar with the technology-driven, let's say, characteristics of an panel. First, we started with an amplicon-based hotspot approach, very focused, very narrow for smaller questions to be addressed. Shifted technology-wise to a hybrid capture-based technology, where we focused on tumor type specific testings on a certain pre-selected set of genes. And nowadays we shifted to a very let's say broader spectrum of comprehensive genomic profiling, either on RNA or let's say where sarcoma fusion gene detection, for example, or CGP testing for solid tumors provided by OncoDEEP. The different dynamic is being driven by whole exome or whole genomic sequencing, which isn't the main benchmark approach we are currently running in our clinical routine diagnostic setup. So the main focus is the CGP approach. So when we think of our hybrid capture tumor type specific approach, which we previously had, it was more a question of jiggling around with multiple very specific tumor panels to be used in parallel in our routine diagnostic setup. This, of course, does have major disadvantages when you think of automation or streamlining your laboratory workflow. Of course, you have to be capable and in line to the regulatory track decision making which are associated to your testing. You have to keep in mind different molecular alterations. The input material, DNA, RNA, was depending on the tumor type different, and the testing assay workflow and analytics also differed in line to the specific tumor type. And with this saying, of course, also the turnaround times and the hands-on times were different, and the requirement that for each and every of these individual tumor type specific approaches, an individual EQA or certification was to be conducted. So we had decision to be taken to shift from such a very narrow focused tumor type specific testing workflow or approach to a more streamlined, single and hopefully automated testing workflow for pan-cancer genomic profile, with the benefit of driving or streamlining differential diagnostics, questions of druggability or option of clinical trials for our molecular tumor board in a let's say more a streamlined and automated manner and fashion. What are the business cases associated to this dynamic? So first of all, we have to keep in mind what are the stakeholders when it comes to such implementation. So I will focus on at least these two sets. One is the clinical DX team doing and conducting these testings, which I am a part of, and then of course the pitch or the discussions we did have with our chief financial officer. Of course, both parties or stakeholders do share opinions and questions being raised, but in some aspects they also differ. To start with, what were our demands? Of course, like I said, very typical. We start with FFP, origin material, to be analyzed, and we were focusing on a pan-cancer approach to have a streamlined workflow to be set up. We wanted to have an up-to-date biomarker-matched therapeutic selection option for our molecular tumor board, to not having the need of sequential testing multiple times for different tumor questions being raised. Each and every nowadays relevant complex biomarker or signature was supposed to be capable to be detected or analyzed. As I indicated before, we do run Illumina-only systems in our setup. So this was also one of the requirements. While doing all these day-to-day clinical testings, of course, implementation, validation, evaluation of a new kit, a new approach, a new technology does bring the need of field application support from the company, which we are demanding for. And of course, we were looking for a product or an option where not only the wet lab, but also the bio IT and clinical decision support was part of the overall product. So these were the criteria shifting from tumor type specific, very focused, narrow panel, or a CGP a vendor, which we previously used, to a new one. Shared questions have been raised concerning the automation. When we did the validation, it was the coronavirus-driven dynamics all over Europe or the globe, and we were raised with a question of automation due to people not being around the lab whatsoever. Reasonable turnaround times is of course always critical when it comes to our molecular tumor board and the clinical demands, as we have a patient history or patient path to follow when it comes to diagnostics, treatment decisions being made. And of course, European regulations, especially IVDR, which has to be kept in mind when we implement, evaluate, or purchase a novel products or testing platforms. Of course, we had a pitch with our chief financial officer and the controlling department, and they raised different questions, which are, of course, also relevant for us, but maybe not the driving force when it comes to our decision making. So they requested certain aspects to at least be evaluated. The first was the requirement of investment. Being comprised of laboratory infrastructure required additional staff or the sequencing platforms. The second relevant issue being raised was the running costs, revenues, which we could expect or which would be kind of modified or evolving in line to our CGP switching from vendor A to vendor B, for example. Especially for the German market, we have multiple networks and guidelines to keep in mind, especially when it comes to reimbursement. So there are requirements and standardized frameworks which you have to adjust if you want to be part of these networks. In Germany, we have the German Network of Personalized Medicines with their own setup frameworking, which was raised the questions if we were capable to adjust by the implementation of this kit to these requirements. And then, of course, scalability and future perspective. If I recollect the pitch, the question or the example was being raised that if we, in the first place, have the sufficient numbers of testing volumes let's say, introduced to our laboratory, because they didn't want to have the situation that, just like we are familiar with, that you do a washing process for a single pair of socks. So they were very keen on the future perspective of these implementations. So let's keep an eye on the financial perspective in the first place. The requirements which have been asked for. As I said before, we were already an established NGS core infrastructure or laboratory with a highly experienced laboratory staff member team, mainly focused on Illumina, Beckman, i7 automation platforms. So this was overall not a major issue, because no additional investment would have to be kept in mind, shifting from the previously used TISO 500 product, shifting to the OncoDEEP product. So this was a very good argument in favor, as the OncoDEEP panel is technology neutral when it comes to the sequencing platform. The same was the case for scalability, as it's possible to analyze simultaneously different batch sizes depending on the NextSeq or NovaSeq sequencing platform. When we check the running costs or revenue, of course, this is very specific to our clinical setup we are working in, and agreements you have with the different vendors when it comes to overall calculations. But this is the scenario we have been working in. And one of the main criteria when we do testing is that we do it in-house. So the money, easily speaking, does stay in-house. It's not a send-out option where the money is being transferred to company A, B, or C, offering their services on the market, and that it's already IVDR cleared. So this was being provided or guaranteed by OncoDNA. At the same time, when you do your calculations on running costs and potential revenues, it's pretty easy, because the wet lab reagents, the bio IT, and the clinical decision support was all part of the product you are purchasing. So there isn't any additional besides DNA, RNA extraction, for example, besides costs which you would have to keep in mind considering this product. Then, of course, it's a pan-cancer solution, so you can streamline and standardize your lab routine workflow independent of the tumor type, which does make sense when you have certain numbers of testings per week to be harmonized and to be scheduled in your routine workforce. And when it comes to efficient laboratory management, overall, at least in our scenario, it was very cost efficient compared to the TISO 500 one for multiple reasons. The striking ones were, or indicated over here. The first, as HRD is a critical biomarker, which nowadays is requested for almost each and every molecular tumor board patient in our clinical setup. And with the previously TISO 500 package or solution, there's always the need of the HRD spike-in, which is then, of course, increasing costs and raising the bar of your overall revenue. Something which is also very striking is that comparing these two options with the OncoDEEP kit, an increased number of samples per flow cell for multiple technical reasons could be batched in a single NextSeq or NovaSeq workflow. So this is also very advancing the overall running cost calculation. And just like indicated before, as it is an all-in-one one-stop solution, including already the bio IT and the clinical interpretation reporting. This is a major difference to the TISO 500 offerings, which were connected at the time we were using it to the offerings of PRNDX, which is to be discussed in a different manner. When it comes to specifications, without going into much detail, the backbone of the library prep is a Twist bioscience technology background, which is in our hands very handy because it's a very streamlined workflow, independent whether you have a DNA, RNA, whole exome, whole genome workflow. It's very comparable when it comes to automation. The DNA input quantity is down to 30 nanograms, which is sufficient for our setup. And the most striking numbers is indicated here on the right, that with a mid-size output scale, you're capable to sequence or batch 8 samples in a single run, which is higher compared to the TESO 500 setup. And this can be increased to a high output cartridge scale of up to 24 samples, or the NextSeq 2000 with a different flow cell. So in case of your individual setup, OncoDNA is offering a cost calculator online. You see the link down here, where you can easily select your preferences or your specific setup criteria, and then you can have a easy to read and easy to use comparison between these two options on the market when it comes to the costs. So these are the required investments, which have not been relevant for us as we are an established NGS core infrastructure. The running costs and revenue were evaluated as very positive as we reduced our costs for running these CGP testings. Requirements by the international networks or guidelines are addressed over here. In Germany, as I mentioned before, we have multiple dynamics when it comes to the option of reimbursement. There is the network of personalized medicine, for example. If you're part of these networks, which were the first one, is relevant for all these overall coverage of a patient DX in the current German healthcare landscape. And we were the first Department of Pathology in Germany to run the OncoDEEP panel in routine diagnostics. Shifting now from the perspective of the chief financial operator or perspective to the lab technical question being raised here, just to recapture our day-to-day demands on the multiple scale of alterations we have to be capable to screen or test for. Of course, this is a rapidly evolving panel of biomarkers or alterations to be screened, and this is just by looking at it already outdated, just as an indication of what are our high and quickly evolving demands on a day-to-day routine setup. So we defined, of course, the nature of multiple biomarkers or alterations to be screened. You have your SNVs, your indels, your fusion genes, CNVs, or the complex biomarkers or signatures. In an ideal scenario, these are all simultaneously being addressed and tested in one workflow. Historically, of course, you can use a single biomarker testing as we all did previously, but this is not just only extending turnaround times and time intense, but it's also very, very insufficient when it comes to the prestige biopsy material or sample material, which is available to us as a clinical setup. So rebiopsying a sample due to a single biomarker testing in a sequential manner isn't an option which we frequently choose. So we adjusted in the past our approach to a multiplex hotspot approach, but of course you can't cover all these relevant biomarkers, and only by implementing a CGP workflow, which we did previously with TESO 500 and now with OncoDEEP, all these simultaneous or different categories of alterations and biomarkers were capable to be simultaneously to be identified. If we compare the two options, it's very obvious that the OncoDEEP Kit is the let's say the improved and most updated panel compared to TS500. It does comprise almost 640 genes, so the number of genes is increased. It's kind of updated depending on the clinical needs. HRD is already included, so there's not the need of an HRD spike in, which has previously been the case or is still the case for TS500 users. And if you check in detail the genes listed down here in green, and if you identify a gene which is relevant for your individual clinical setup, which is indicated here in green, these are the differential genes which are not covered by TS500. The only gene which is missing if you merge the TS500 panel design and the OncoDEEP design is the TERK gene, which is, from our perspective, not relevant for clinical routine. The grayish highlighted genes or names are just older name annotations being modified or updated by time. So just by indicating or just by the indication provided here, you see the content or the gene number is highly increased, HRD is included, and the striking part is that the increase in gene numbers is being conducted even by lowering the genomic content needed to be screened. And this is the reason, from a technical point of view, why you are capable to sequence a larger batch size or sample size compared to the Illumina TS500 design. Just as an indication to our timeline or overall workflow, before we shifted to fully automation, we had a 2.5 to 3 day library prep, which is being reduced to 1.5 day workflow. Usually we start on Tuesday until Wednesday with the automation. On the NextSeq scale, the turnaround time or the sequencing duration is less than 24 hours, being conducted on Friday or on Saturday. And bio-IT, or the secondary and tertiary analysis, is being conducted over the weekend. This is very handy for us, that the let's say bio-IT is being initiated and is running over the weekend, so that up to a time scale of 48 hours on Monday, latest on Tuesday, we have the results for our internal evaluation, interpretation, and the final reporting. So overall, in an ideal scenario, when nothing is going wrong or no QC check failed, and we have a turnaround time of 5 working days. Automation, like indicated before, is pretty streamlined. We only have 2 intervention points for QC and pooling reasons. There's no rocket science behind the automation. It didn't take us longer than 4 to 6 weeks with the support of Beckman Coulthard, Twist, and field application specialists from OncoDNA. So they were all 3 very helpful in implementing this new to be shared or to be also discussed protocol for the automation, followed by the sequencing or the analytics. Without going into much detail, just for calculations, since end of 2023, we started routinely using this CGP panel. In the meantime, we have more than 180 library preps being conducted, counting for more than 1,600 samples in total. Per average, we have 2 to 3 library preps being run per week, with an less than 3% of failure rate, most of the time due to the low input DNA concentration or low input FFPE, let's say, prone errors, which we are all aware of due to fragmentation, for example. When it comes to such an automated and streamlined workflow, of course, reflex testing is an issue, and we decided to come up with a reflex impulse for the testing for at least these 6 tumor entities, independent of the clinical stage or the status of metastatic disease or relapse whatsoever. We do have a relapse testing for these 6 tumor types being established. Just by checking the overall library prep quantity and quality, we do generate very robust and high quality poolings and final library pools for sequencing. That's an average scale or data output from one of our very representative automated workflow, which we do generate on a weekly basis, which is within the framework or the requirements being provided by OncoDNA. One of the last issues, of course, besides the technical questions being raised or needed to be addressed from our perspective, coming from the laboratory, was the evaluation and validation. We had a special implementation workflow or protocol comprising previously tested samples to be reanalyzed with this automated or manually processed or tested samples, where we are aware of the profile to be identified. We also checked quality controlled reference material in parallel, knowing the profile and what to expect, what to screen for, so you could easily compare the profile as well as the allele fraction being detected. And of course, we applied and conducted external quality assessment testing schemes comprising HRD testing, a multi-gene scaled approach with a panel larger than 1 megabase. And we had a lung cancer related marker EQA successfully conducted, comprising Alk-ROS-RET fusions and MET exon 40 skipping, these all being done on DNA level. And the latest was the BRCA1/2 EQA, which we successfully participated in. In the last 2 weeks, we just received EQA samples for these grayish indicated samples, but we are very positive, and the first sequencing results are also positively, that we are optimistic to also succeed in these EQAs. Only focusing on HRD as an example, just to highlight the performance over here. Like I indicated before, we used a commercial Cereseq provided reference material. And just as a quick recollection, the recommendation input provided by DNA is a minimum of 30 nanograms. We did it in dilution steps, and even going down to 4 nanograms in our scenario, we could see that for this specific sample, the overall, let's say, our HRD scoring and calling was very robust and stable, even by lowering down the input material down to 8 nanograms. So this was very striking, very promising. And we also conducted the EQA for HRD, where we did receive 10 ovarian cancer samples, FFPE material origin. The standard was the My Choice Myriad Genetics scoring. We had 15 working days to succeed, and the striking readout was that 20% of participating laboratories failed either by providing false positive or false negative results, which is for a clinical setup, of course, critical. Based on our expertise and sequencing, we succeeded for all of these 10 samples being correctly analyzed. So this was a very nice indication that comparing all these competing vendors on the market, that OncoDNA or OncoDEEP was sufficient, at least in our hands, to succeed in this EQA for HRD detection. To finally summarize before shifting into the discussion, I want to highlight the key benefits from my personal point of view, comparing OncoDEEP to the panel or offerings we used before. We have at least three categories to focus on. The one is the financial aspect, the productivity and the quality. I go through these in detail. So the first take-home message by our evaluation and day-to-day practice and usage is that we are capable, just by using the OncoDEEP Kit, to reduce costs per sample. This is being guaranteed or funded by that it's an in-house all-in-one solution, where the kit does comprise the wet lab reagents and the bio-IT, which isn't being separately and cost efficient. Then, of course, it's a pan-cancer approach where we were capable to streamline our laboratory automation. We could reduce hands-on timings in our laboratory infrastructure, and this is the most reasonable one. As a financial argument, there's no need for the relevant HRD cost intense spiking required anymore compared to the TSO 500 one. And when it comes to the calculation, an increased number of samples per flow cell to be sequenced simultaneously. Just as a recollection, you can check it on your individual scale or for your individual setup with the cost calculator provided by OncoDNA. When it comes to productivity, just as take-home message for our setup, we did highlight an improved turnaround time for our molecular tumor board. Of course, this is very important for the, let's say, value chain coming from FFP material, going through library prep, sequencing, and the time until you have the clinical interpretation and the reporting finalized. So we could speed up the whole process due to automation and less complex twist-generated library prep. We had improved scalability due to the increased batching of our sequencing approach. The more samples you can simultaneously sequence, the faster you're getting, and the integration and the harmonization of the already included and harmonized clinical decision support, the QC, and reporting provided by OncoKDM as the clinical interface for the bio-IT solution, was compared to the peer-in-the-X, formerly available, only available peer-in-the-X option, far superior in our comparison. And the last one is the quality. Of course, as we have a clinical pressure of being innovative and being up to speed with the clinical demands when it comes to clinical decision making in our day-to-day diagnostic testings, the profile of the panel or the kit itself is the most updated biomarker matched therapeutic selection option on the market. In our evaluation, TSO 500 is just lacking sufficient gene sets which we are depending on to be screened for. HRD is included. Based on our FFPE origin of the majority of samples, we could generate and secure a robust and very sufficient library prep workflow with a high sequencing quality being generated. And we also succeeded in our participation of multiple EQAs, which is relevant for our certification and laboratory clearance, which is being looked at from a regulatory and guidance perspective. So we had multiple biomarkers screened and established based on the OncoDEEP scenario in our workflow. With this said, thanks very much for your lookout. I'm looking forward for your questions, and let's keep in mind, keep on testing, because unless we are screening for novel biomarkers or established biomarkers, we won't be able to identify patients subpopulated to benefit of whatever therapeutic option is available. In the end, always happy to help, so get in touch and let's see what questions are being raised. Thanks very much.
Thank you, Marcel, that's really helpful. And while we're just inviting questions, because I don't see any yet that have been flagged, but I'll just wait for some of those to emerge. I'd like to ask you a couple, if you don't mind. So in terms of productivity, you mentioned 5 working days that you're working to. Is that something that you've set internally, or is that something set nationally through your precision medicine network, or what are the influencing factors on turnaround time?
No, that's a benchmark we did came across depending on the workflow. So we didn't have the matching threshold of 5 days set up by ourselves or by any guideline we have to face, because this is very, very fast and very straightforward. This is just the established workflow we're working in. So 10 to 14 working days is the benchmark. In Germany, we have to address these kind of testings. So 5 days, even having enough time for repeated testing, once you have a secondary DNA isolation, for example, being needed due to failure to QC, for example, is still capable to be done in this 5 days turnaround time schedule. But this isn't any adjustment based on a national-wide or international-wide guideline. Okay. It's just due to the very straightforward automation and easy to use twist backbone workflow.
So Marcel, does your library prep approach make use of unique molecular identifiers?
No, it doesn't. It's not, but this is a limitation based on a twist library prep. So that's nothing which is on OncoDNA side. But we are not using unique molecular identifiers in the current scenario.
Okay, and what provider do you use for your EQA controls?
We have at least 2 options in Germany. One is called QUIP. It's a German, let's say, company organizing and monitoring these kind of EQAs, and there's a European approach from the European Society of Pathology. Sometimes they're partnering up. I think the lung cancer related marker EQA was a pairing of the QUIP and ESP organization.
Okay, thank you. And sorry, just coming back to your validation as well, which is kind of linked to that. You did an internal validation. Correct. What were your numbers for that, and who kind of dictated that? Did you set that for yourself, or did you use an external kind of, shall we say, influence to decide on the number of tissues that you would validate?
In Germany, there's not a clear cutoff or a minimum number of samples you need to analyze during your validation. It's something like being stated as sufficient number of samples, which could be 10 to 500 in the long run. From our perspective, as we do validation and implementation for multiple testings as a routine procedure per year, we have the scenario that we do multiple dilution steps, we do multiple application by different staff members. We do previously EQAs where we have the samples still available, for example, and we have a certain set where we are aware of which have been critically and previously, let's say, workflow to be tested, larger indels, internal tandem duplications, which are known to be tricky, which are tested. So I would say in the long run, we had, by the time speaking, at least 200 validation samples being processed and analyzed in parallel. Okay. Or in total, not in parallel, in total.
That's really interesting, thank you. A lot of people ask me that question, and there's loads of questions coming in now. So there's actually a group of questions about reimbursement. So some of these are about particular markets, which I don't think we'll get into right now, but we can talk about that outside of the meeting. But I know that OncoDEEP is supported both by private health insurers globally, I mean, and by, shall we say, centrally funded entities. Within Germany, how does that work? How does the reimbursement work for your lab?
That's, of course, on a country scale different,
exactly.
depending on your regulation and guidelines, even when it comes to different medical infrastructures. In Germany, we have the dynamics. It's called Modellvorhaben genome sequencing, where it's a high demand. It's a pilot study for a period of 5 years, where coming from CGP going to by time to the exome or the genome. And in this network or in this pilot study, there is reimbursement being secured, but also tumor type specific. When there's a therapeutic indication for, let's say, HRD testing, then the reimbursement is secured and can be requested for. So in our scenario, it's funded and it's on an individual scale also reimbursed. Yes.
Okay, and as we know, that's a very complex discussion for individual markets, and maybe that's something we need to pick up in a future user group. Somebody's asking here that you mentioned OncoKDM decision support tool. Do you want to talk a bit more about that? How was that for you in your work?
I mean, it does make our life easier, right? Basically, it's an interface where you do get a list of all the, or the profile, the genomic individual profile is being illustrated with different dashboards indicating the biological impact, the annotation on DNA, protein level, the exon being alterated or being involved. You have a link to potential druggable targets or therapeutic intervention options for the molecular tumor board. So it does make our life easier. It has a comprehensive summary being provided, so that you, of course, we still have and we still conduct interpretation on our own scale, but doing these standardized or addressing standardized questions like, is it a pathogenic disease alteration? Is there already approved drug in association to this individual profile being approved? What are potential clinical trials? This is all being provided automatically in the OncoKDM option, and this is very handy because it's very time sufficient in our hands, in our clinical setup.
So I would say you're like many of our customers, that you're using that in combination with your own local, shall we say, interactions with the data. But you didn't call it out as a point of improving productivity, that bit of reporting. But would you say that going forward you could do that you could improve the way in which you're reporting enabled by KDM to drive further productivity in your teams? Or would you say for now that's not where you would want to go?
No, of course, it does drive improvements when it comes to the time spent for the interpretation on an individual patient perspective or scale. You have to keep in mind we were coming for PRNDX, which is a kind of black box. You do receive an automated report in a PDF formatted scale without having the option to, let's say, explore the overall profile of the patient. So you just had the report, which wasn't sufficient from our demands because you didn't see what kind of thresholding was applied or what else was there from a variant of unknown significance perspective, for example, which wasn't called in the clinical reporting. And when it comes to this, just to the sample and testing volumes, of course, it's a question of expertise, but sometimes you have a profile which is being discussed in a molecular tumor board where you still have to do research on the, let's say, background of the gene or the protein itself. And this is also already provided in the oncology spectrum by OncoDNA. So you have a shortcut profile indicating what is the biological relevance and let's say transformed consequence of an alteration in whatever the protein is. So this is very handy to have a first indication of what kind of signaling pathway or therapeutic path could be discussed. And this is all in one interface, so you don't have to do, of course we're doing it, but as first indication, you don't have to do your in detail PubMed research upfront. You can stay in the interface and have first line indication or information being provided.
Okay, that's really interesting. Thank you. I think what's coming through on some of these questions, Marcel, is that obviously everyone starts at a different point, right? So you were really lucky in your lab. You were already working with a lot of kit. You've got many of the processes in place. You'd work with a legacy panel, TSO 500, which you called out. There's somebody here on the call today that's saying that you know, they would be starting from scratch. And so could you explain the process from the FFP block to the library prep through to the use of the kit? And I mean, you mentioned in some way how you kind of decreased the amount of time to those steps. And you did call out at the beginning what instrumentation and things you had. But, you know, for somebody like that, starting from absolute scratch, what would your advice be? And would you be able to perhaps, if not today, call out those steps to help them in a more detailed way, Marcel?
So the good thing is it's basically not rocket science. So you don't have to be cautious or anxious in using this kit, because once you are already experienced NGS user, it's easy adaptation. If you don't use NGS in the current scenario, it's also not a very headache concerning approach. So you start with your already established FFP block sections being sliced. You have your DNA or RNA extraction depending on the workflow, which is automated or manually being conducted. Then we have first QC checking tumor cell content and certain parameters based on morphology or histology, immunohistochemistry, staining. And then it's a standard procedure in the laboratory where you start based on your DNA, depending on the concentration and quality of your DNA, processing with the library prep. This is very streamlined, independent of the automation, because you have certain master mixes being pipetted, being incubated, being shaked, being size selected, whatsoever. But this is nothing which is critical from an unexperienced perspective. When it comes to sequencing, it's the same. Of course, you need to kind of have the experience of balancing out your poolings, your sample batches to be run simultaneously on a Illumina sequencing cartridge. But this is also a question of expertise and advice being provided by Illumina. So I wouldn't argue in favor of not implementing such an approach just by the fact that you haven't done it beforehand. So it's easy to do. It does take, for certain setups, longer than for an experienced setup, but I would argue in favor that each and every laboratory who's capable to isolate DNA and has a manual experience with pipetting is capable, as long as there's a sequencing infrastructure available, is also capable to do this really easy hands-on workflows.
Interesting, thank you. And the thing that's been interesting for me and my customers in the UK is that because we've used the OncoDEEP centrally, for those that have wanted to go, if you like, through a step change, we've been able to help them by using OncoDEEP within our own lab, and then when they're ready, transition it back. So they've bought the kit, used it in our own lab, built up their learning and capacity to make the step change, and then moved the testing into their own lab, and we've held their hands to do that. Just coming back to the validation piece, though, for somebody like this individual on the call, what would be your advice around validation? Because obviously that's a very important step in terms of making that change.
Of course, it's very critical, but independent of the assay or testing or workflow you're trying to implement for clinical testing, because in the end there's a patient history of therapeutic decision making in association to it, you have to make sure that you're aware of the strengths and the weaknesses and the limitations when it comes to sensitivity, specificity of your testing workflow. So you have multiple approaches to identify a fusion gene, for example. You can do it by immunohistochemistry based on staining patterns, cellular expression whatsoever. And it's the same with an NGS approach. If you are not aware of the limitations in line to a specific genomic region or a specific alteration due to the size of an deletion or insertion, for example, or complex rearrangement, then you, of course, face the troubling of a false negative or not false positive, but false negative result for patient from patient characteristics. So knowing your strengths and weaknesses is important, but here you have an IVDR cleared product already on the market or being launched. So it went already to a certain level of validation, which is important, independent of the sequencing, but because it's technology neutral, you can do it on whatever platform you want to use it for. But there has been extensive validation already being conducted. Not saying that I would always recommend each and every laboratory to do an in-house validation just to gain these needed expertise, depending on your certain let's say frameworks or different levels of input materials you're faced with. In our scenario, we could see different levels of FFPE archived samples being processed depending on the origin where we do receive the samples from in the first place. And this is something you have to be aware of when you're using these complex testing kits or assays to know the limitations.
And certainly with our field application specialists, we have implementation plans. You were one of our earlier customers, so perhaps our plans weren't as robust then, but we like to hold the customer's hand with a structured project plan, so to speak, that takes them through those steps. We're coming up to the end of our time now, Marcel, and thank you so much for sharing all of your insight. But I would like to leave you with one question. So you mentioned those stakeholders that you had to convince, your terrible financial controller.
They're not so terrible.
Not so terrible, I know. But nevertheless, did you have to create a written document? And if so, would you be willing to share some of the, shall we say, segments of that, not the real detail for your lab, just to help somebody who was starting from the very beginning prepare a business case?
No, we didn't. We, of course, had an internal pitch where I did a presentation. What is the current state? What are the current costs? And what are the to-be costs, perspectives, and the future trends by implementing or shifting from vendor A to vendor B, for example. And this is easy to do. I mean, you just have to list your current costs, and then you see the costs for your specific setup or scenario, depending on the kit you're using. The cost calculator being provided by OncoDEEP is very handy. And the two key factors to keep in mind is no HRD spike in required, so you can erase these costs already from your calculation, and the batching size is improved, so the readout per, depending on the cartridge size, is higher compared to the, at least in our scenario, previous scenario, and these are the two driving cost factors, at least in our calculation.
Okay, so really using the cost calculator, preparing a presentation allowed you to facilitate that conversation then in your organization. Yes. And are they holding you to account? Are they measuring, you know, your changes that you said would result from the improvement in moving to the OncoDEEP kit, or are you just monitoring that yourself?
Of course, we do monitor it. We don't report it to the controlling anymore because it has been approved. We of course have our internal, from the other side of the stakeholder, which is the molecular tumor board, the requirement of even getting faster, coming from 5 days turnaround times to be even more faster, improving and generating even more information. But we already have a very high standard level of quality, I would say, in our routine setup, which is of course specific to us, but that's nothing which can't be achieved by each and every laboratory, I would say, independent if you do it manually or automatically. And in case any questions being raised, I'm more than welcome to address or to change or to share my expertise.
Thank you, Marcel. And you've left your email address there, so that's very kind of you to share that with the participants today. Thanks again for your input. I'm sure we'll hear from you again. We'll look forward perhaps to hearing your onward journey around OncoDEEP, perhaps at our next user group meeting, which we hope will be in Germany next year. But in the meantime, thank you to everybody for joining the call, and we look forward to welcoming you again on OncoDNA's webinars in the future. Thank you.
Thanks very much. I appreciate it.